Welcome to Binary Bio, the channel where biology meets computation. Modern biology is producing more data than ever before. A single sequencing run can generate terabytes of DNA data. In this video, we'll walk through four important machine learning algorithms that are widely used in biological research. We'll understand the intuition behind them and how they help solve problems like predicting disease, identifying biomarkers, classifying genomes, and modeling gene expression. By the end of this video, you should be able to look at a biological dataset and understand what type of machine learning method might help analyze it. In many biological systems, the relationships between variables are extremely complex. Machine learning instead looks at large datasets and automatically finds patterns that explain the observations. There are three main categories of machine learning. Supervised learning uses labeled data. Each example already has a known answer. For example, imagine we have gene expression measurements from patients and we know which patients have cancer and which are healthy. The algorithm learns patterns that distinguish the two groups and then predicts the label for new samples. Unsupervised learning is used when the data does not have labels. The algorithm tries to discover structure within the data. For example, in single cell RNA sequencing, we may measure expression of thousands of genes in thousands of cells. The algorithm groups similar cells together, which often reveals different cell types. Reinforcement learning is a different paradigm where an agent learns through trial and error by receiving rewards or penalties. This is sometimes used in problems like drug discovery or molecular simulations. Across all machine learning methods, there is a fundamental challenge known as the bias variance trade off. If a model is too simple, it cannot capture the real patterns in the data. This is called underfitting. If a model is too complex, it may memorize noise instead of learning true biological signals. This is called overfitting. Good machine learning models balance these two extremes. Now let's look at four algorithms that are widely used in biological research. One of the most fundamental techniques in statistics and machine learning is linear regression using something called ordinary least squares. Imagine we want to understand how temperature affects gene expression. We measure temperature and expression levels for many samples and plot the results on a graph. Temperature is on the x axis and gene expression is on the y axis. We might suspect that as temperature increases, expression increases as well. The simplest model that describes this relationship is a straight line. A straight line can be written as baseline plus slope times the input variable. The baseline represents the expression level when temperature is zero, and the slope describes how much expression changes when temperature changes. If the slope is positive, expression increases with temperature. Even if temperature influences expression, other factors like transcription factors, cell conditions, or measurement noise also affect the values. So the points on the graph will not fall perfectly on a straight line. Instead, they scatter around it. For each data point, we can calculate an error, which is the difference between the actual measured value and the value predicted by our line. Some errors are positive and some are negative. If we simply added these errors together, they might cancel each other out. To avoid that, ordinary least squares squares each error before adding them. This produces something called the sum of squared errors. Squaring the errors accomplishes two important things. First, negative and positive errors both become positive, so they don't cancel out. Second, large mistakes are penalized much more strongly than small mistakes. An error of 1 becomes 1 when squared, but an error of 3 becomes 9. The algorithm tries different possible lines and calculates the total squared error for each one. The best line is the one that produces the smallest total squared error across all samples. In other words, it is the line that best fits the data. Linear regression is widely used in biological research. It appears in gene expression analysis, EQTL studies that link genetic variants to expression changes, and drug dose response modeling. Now let's move to a powerful classification method called support vector machines. Suppose we want to classify genomic samples into two categories. For example, bacterial genomes and viral genomes. Each genome can be represented by numerical features such as GC content, sequence length, or the frequency of certain sequence motifs. If we plot these features on a graph, each genome becomes a point in space. Some points belong to bacteria and others belong to viruses. Our goal is to draw a boundary that separates the two groups. Many different lines could separate the data. Support vector machines choose the boundary that maximizes something called the margin. The margin is the distance between the separating boundary and the closest points from each class. Those closest points are called support vectors. You can imagine the margin as a safety buffer between the two groups. A larger buffer means the boundary is less sensitive to small variations in the data. This is important because biological data often contains noise. By maximizing the margin, SVMs tend to produce models that generalize better to new unseen samples. Sometimes the data cannot be separated with a straight line. Imagine two groups shaped like concentric circles. No straight line can divide them. Support vector machines solve this using a technique called the kernel trick. Instead of drawing a boundary in the original space, the algorithm transforms the data into a higher dimensional space where separation becomes possible. A pattern that looks tangled in two dimensions may become separable in three or more dimensions. This idea allows SVMs to model surprisingly complex relationships. Support vector machines have been widely applied in biological problems such as protein structure prediction, cancer classification using gene expression data, and genome annotation. Next, we look at random forests. Random forests are based on decision trees. A decision tree makes predictions by asking a series of questions about the data. Imagine a doctor diagnosing disease. They might ask questions like: is protein A above a certain level? Is gene B expressed? Is the patient older than a certain age? Each question splits the dataset into smaller groups. The goal of each split is to separate the data so that the resulting groups are as homogeneous as possible. In classification problems, this means each group ideally contains samples belonging to the same class. Decision trees measure the quality of a split using metrics such as Gini impurity. Gini impurity measures how mixed the classes are within a group. If a node contains half cancer samples and half healthy samples, the impurity is high. If a node contains only cancer samples, the impurity is low. The tree chooses the split that reduces impurity the most. By repeatedly splitting the data, the tree eventually creates branches that lead to final predictions. However, single decision trees have a weakness. They are extremely sensitive to small changes in the dataset. If the training data changes slightly, the structure of the tree can change dramatically. Random forests solve this problem by combining many decision trees together. Instead of building one tree, the algorithm builds hundreds of trees. Each tree is trained on a random subset of the data and a random subset of the features. This randomness ensures that each tree learns slightly different patterns. When making a prediction, every tree in the forest gives its vote. The final prediction is determined by the majority vote of all trees. This averaging process reduces variance and produces more stable predictions. Random forests are widely used in biological research for biomarker discovery, identifying important genomic features, and predicting disease outcomes from high dimensional biological data. Finally, we arrive at gradient boosting, one of the most powerful techniques used in modern machine learning. Unlike random forests, where trees are built independently, gradient boosting builds trees sequentially. Each new tree focuses on correcting the mistakes made by the previous trees. The process starts with a very simple model. For example, the initial prediction might simply be the average value of the target variable. Next, we calculate the residuals, which represent the errors between the predicted values and the actual values. These residuals show where the model is performing poorly. A new decision tree is trained to predict these residuals. In other words, the tree learns patterns in the mistakes made by the current model. Once the tree learns how to predict the errors, its predictions are added to the original model as a correction. The updated model becomes slightly more accurate. This process repeats many times. Each new tree adds a small improvement. Instead of building one large complex model, gradient boosting builds many small models that gradually improve the predictions. The term gradient comes from optimization. The algorithm calculates the direction that reduces the prediction error most efficiently and moves the model in that direction step by step. One of the most widely used implementations of gradient boosting is XGBoost. XGBoost adds several enhancements such as regularization to prevent overfitting, efficient parallel computation, and better handling of large datasets. Because of these improvements, XGBoost has become extremely popular in machine learning competitions and real world data science. In biological research, it is used for predicting disease severity, modeling drug responses, and integrating multi-omics datasets such as genomics, transcriptomics, and proteomics. Each of the algorithms we discussed has different strengths. Linear regression is simple, interpretable, and useful for modeling relationships between variables. Support vector machines are powerful for classification problems and work well with high dimensional data. Random forests are excellent at capturing complex feature interactions and identifying important variables. Gradient boosting methods often produce extremely accurate predictive models and are widely used in modern data science. Choosing the right algorithm depends on the biological question and the characteristics of the dataset. If you enjoyed this video and want to learn more about the intersection of biology and computation, consider subscribing to Binary Bio.